The field of ultrasonic nanomedicine can conditionally include work in which the interaction of ultrasound and small particles, such as micelles, liposomes, nanoemulsions, bubbles, and nanocapsules, is used as the main factor, regardless of their nature. The size of these particles ranges from 10 to the minus ninth to 10 to the minus sixth meters. They are introduced into the body in a ready-made form, that is, systemically. As a result, the effect of ultrasound on a tumour containing aggregates of nanoparticles can lead to inhibition of tumour growth and, in some cases, to complete remission. The method of ultrasound nanotherapy for malignant tumours that we are developing differs from those described in the literature in one important respect. Nanoparticles and their aggregates are formed directly in the tumour from a non-toxic and non-medicinal substance injected into the bloodstream as a solution. The formation of nanoparticles and their aggregates directly in the tumour is ensured by its metabolic atypism. This includes a low pH, an increased content of calcium ions in the intercellular fluid, and the monotony of the lipid structure of the membranes. The solid phase is released into the tumour after intravenous administration of solutions of compounds whose calcium salts or acidic forms are insoluble in the tumour. The parameters of atypism can be controlled systematically. For example, an increase in glucose in the diet leads to acidification of the tumour, which facilitates the formation of a solid nanophase within its volume. Comprehensive preclinical studies on the therapeutic efficacy of ultrasound nanotherapy for malignant tumours have been conducted at the N.N. Blokin Russian Research Center in collaboration with the Faculty of Chemistry of Lomonosov Moscow State University. The studies were performed on different types of animals, both healthy animals and animals with transplanted tumours. The experiments were conducted on prognostically significant solid tumours that developed in the paw muscles of mice, Lewis lung carcinoma or LLC, melanoma, B16, cholangiocellular carcinoma or liver cancer, PC1, and adenocarcinoma, CA755. Ultrasonic exposure was performed at two frequencies, 0.88 MHz and 2.64 MHz. The total intensity did not exceed 5 watts per square centimetre. The animal was fixed on a special plate and placed in a thermostatically controlled bath at a temperature of 36 degrees Celsius. The exposure time was 10 minutes. The effectiveness of treatment was quantified using data on tumour growth dynamics in the control and experimental groups. The animals were ranked into groups with the same tumour volume. The volume of experimental tumours varied from 1 to 2 cubic centimetres. After ultrasound exposure at certain time intervals, the average tumour volumes in the experimental and control groups were calculated. From the dependence of experimental tumour volume relative to control volume on time, the time required for doubling of the tumour volume was determined. Theraphthal, the octasodium salt of octa-4,5 carboxyphthalocyanine of cobalt, developed by SSC Niopic in Russia, was used as a sonosensitizer. Its calcium and acidic forms are insoluble in biological fluids at pH below 5. Hydroxyapatite nanoparticles coated with albumin were also used. In this case, albumin acts as a natural vector, binding hydroxyapatite to the tumour. The figures above show examples of tumour growth dynamics under the combined action of ultrasound and Theraphthal, and ultrasound and hydroxyapatite. In all cases, there is a slowdown in tumour growth. The doubling time of the tumour volume increases many times. The life expectancy of the animals is also increased by 30 to 40%. No toxic effects were observed. When cytostatics are included in the treatment scheme, in some cases it is possible to achieve complete remission of the tumour. The enhancement of the therapeutic effect produced by the introduction of nanoparticles is due to a decrease in the cavitation strength of the medium. A decrease in cavitation strength also occurs with an increase in the gas content of the medium. This can be achieved by acidifying the tumour through systemic administration of glucose, followed by administration of sodium bicarbonate. This leads to a noticeable anti-tumour effect. Histological and electron microscopic studies have revealed that ultrasound in the presence of Theraphthal causes extensive damage to blood vessels, plasma membranes, mitochondria, and other tumour cell organelles. An immunohistochemical study revealed that ultrasound in the presence of Theraphthal has an inhibitory effect on neoangiogenesis. These changes are observed after 3 hours and intensify over the following 48 hours. Conclusion. The choice of the optimal technique for introducing nanoparticles into a tumour is determined by specific conditions: the type of tumour, its location, the comparative toxicity of the nanoparticles, the duration of the procedure, and other factors. In our opinion, the method of synthesizing nanoparticles directly in a tumour may in some cases be simpler in clinical implementation and less toxic. With favourable development of the theoretical, experimental, and instrumental basis, this method should complement existing methods of cancer therapy. Special attention should be paid to the development of systemic methods of non-cytotoxic supportive therapy.